Thanks again for having me here. And I chose vitamin D and BMI. And vitamin D as it relates to genetic variants. And the reason I chose this is I wanted to talk a little bit about the work our laboratory is doing. And I'm going to cover a lot of ground in a few minutes, but I'm available to answer questions. But one of the things that I wanted to point out is a lot of times when we do genetic association studies, we always control for sex. And we don't really think about looking at men and women separately. We do in epidemiological studies, but not for genetic association. And it caused me to go back and look at some of our work when Nina called me about this meeting. And we have some interesting findings I'm going to share with you today that aren't published yet, and we're looking right now, we're replicating them to see if we can find some. Okay. Most of you already know that age-related macular degeneration is the leading cause of visual disability, not just in the developed world, but we're seeing it a lot in the developing world, especially in Nepal when I was just there 2 years ago. And recently, a meta-analysis was just done that actually replicated the findings from over 10 years ago and found that females are 1.13 times more likely to develop age-related macular degeneration than males. So that could be because they live longer, they may have a different set of risk factors, we really don't know why. And there's a need to get to that level of mechanism. It could be hormonal, we don't know those answers. But the thing is, as the population continues to age, AMD will be a more common cause of vision loss than diabetic eye disease and glaucoma combined. Next slide. Now, you've all heard of environmental factors that affect age-related macular degeneration that increase risk. But these are the same environmental risk factors that are associated with a lot of diseases that have at their root complex inheritance: cardiovascular disease, diabetes, cancer. The thing about age-related macular degeneration, we already know there's two major risk genetic loci. You've heard of the CFH, complement factor H, and the chromosome 10 loci, which we don't really know which gene it is. Is it ARMS2, is it the H trial loci, or are they just one gene? Recently, you all may have seen our Nature Genetics paper that was done by 18 groups, published in Nature Genetics just a couple of months ago. It was a major undertaking that found 7 new loci by a large genome-wide association study. And that was 57,000 people that were studied, cases and controls. And we're now doing an exome chip study to find rare variants associated with age-related macular degeneration. And our group, as well as a lot of groups, are part of that study. Also, we have modifier genes that are likely going to be representative of drug targets. And our group, Nina Hadar, has done a lot of work with us on this, is RoboLaura and a vitamin D metabolism gene, which I'm going to talk about today. Next slide. A study approach that we've utilized, and I started as a postdoctoral fellow at the Massachusetts Eye and Ear Infirmary with Joan Miller and Ivana Kim, was doing family-based studies of age-related macular degeneration. And that was using a SIV-based pair approach because this is a late onset disease. Parents aren't available for study, and the children are too young to manifest the disease. The thing about using the family-based approach is that you're getting rid of all the noise, the false positive discoveries that are associated with genetic associations in complex diseases, but also each member of a pair are already matched for race and ethnicity. And that can be problematic with discovery studies. because you'll see these studies, especially with complex diseases, where people can't replicate findings. So this is what we've always used as discovery. Next slide. And the thing is, and you just heard from David Epstein, which we've been using for years, is a systems-based biology approach. We use epidemiological data, good, robust phenotypic data, expression data, and genetic data to get to our answers or our findings. And I'm going to show you an example of that. Oh, next slide, sorry. And that was one of our first findings, where we showed, and Dr. Hadar, Dr. Schomburg, who I saw a picture of earlier, was also part of this. And we showed that this is the first study actually that showed a genetic basis for vitamin D metabolism in association with vitamin D risk. With age-related macular degeneration risk. And we showed that having bad variants or risk variants in some of the vitamin D genes increased your risk of all types of age-related macular degeneration. Next slide. And this wasn't far from what other people had shown, that showed that having low levels of vitamin D also increased your risk of having early age-related macular degeneration. Next slide. And having low levels of vitamin D has been shown with having risk of hypertension, autoimmune diseases, and osteoporosis. And vitamin D also functions in mechanisms that have also been shown to have overlapping mechanisms with age-related macular degeneration, like apoptosis, inflammation, and angiogenesis. Our ability as we age to synthesize vitamin D is also impaired. As we age, our ability to synthesize vitamin D is reduced by 75% over the age of 75 years. And most people, I don't know if you get your vitamin D checked. In Boston, usually, it is considered a standard of care. In Utah, it's not. You have to request it. But most people don't have enough vitamin D and do require vitamin D supplementation. Okay, and vitamin D and body mass index in general, a few studies have just come out within the last year, and within just 2 years ago, that specifically looked at women and found that variation within the same gene that we showed, the CYP24A1 gene, which is the major catabolizing gene of the vitamin D pathway, was actually associated with body mass index and weight in women, and actually impaired, that people were at greater risk, people who were overweight, there's an inverse relationship between vitamin D and body mass index. It's thought that being overweight impairs your ability to metabolize vitamin D. And then here, serum vitamin D is inversely associated with body mass index. And then several other studies have shown this as well. Next slide, please. So we wanted to look, and also in age-related macular degeneration and body mass index, Johanna Seddon, who I know is on the advisory board for this committee, has shown in both men and women that higher body mass index is a risk factor for age-related macular degeneration. And Dr. Schomburg showed that low body mass index and having a high body mass index is associated with increased risk of age-related macular degeneration, but only in men. However, this was the Physicians' Health Study, and that is only a study on men. Next slide. However, in our cohorts, when we looked at both men and women, and these are cohorts, the Leland Sidpier cohort, we have a cohort of a homogeneous Greek cohort from Larissa, Greece, and we have a large Korean cohort from Seoul. We did not find this to be the case in these cohorts separately. However, when we pulled our cohorts in their entirety, we found this to be the case with females only for risk of neovascular age-related macular degeneration. So that increased body mass index was associated with increased risk of neovascular age-related macular degeneration in only females. Next slide. So, we said, how do we look at this whole story based on what the literature is showing for vitamin D and body mass index? And we went back to this old data we had. And we looked at what we found in the major catabolizing enzyme, the vitamin D pathway, that we had already shown to be associated with age-related macular degeneration risk. And this is a schematic of the entire gene that we had studied. And here were the significant single nucleotide polymorphisms that we had found significantly associated with age-related macular degeneration risk. And this is just indicating the 5 SNPs in each region that were shown to be associated with risk. Next slide, please. And we said, let's look at the interaction of these things and what this might mean. And the thing is, even though we have all these genetic associations out there, we still can't explain, in general, a significant portion of the heritability of age-related macular degeneration. And the thing we're still trying to do with the consortium is we're still trying to figure out the best way to study interactions. That is gene-gene interaction and gene-environment interaction. And these are the 2 types of interactions we're trying to explain in genetic studies. And this is known as epistasis. And this is looking at things, and an interaction is defined as, is the effect greater than what you would expect than looking at either factor alone? And how would you study this? Next slide, please. So, we looked at these factors individually. And the thing is, you have to remember, when you're looking at all these statistical interactions and you're running all these tests, you've got to control for the number of multiple testing. And a lot of researchers forget to do this. And lo and behold, when we did this, we found this to be true for all 5 SNPs that we found to be significantly associated with age-related macular degeneration risk. However, we did not find this to be true when we combined the males and females. We only found this to be true in the females and only for neovascular age-related macular degeneration. And this is an example of one of these SNPs. Now, the way we look at this is here are the green bars representing the females with neovascular age-related macular degeneration in the sibling cohort. And these little alleles, C and T that you see here, TTs, the C is the risk allele. And as we see an increase in the number of risk alleles, the body mass index is reduced. It goes down. Here's the body mass index on the y-axis and the number of risk alleles here on the x-axis. And it goes down. We don't see this to be true for the normal female siblings without any AMD. And what this tells us, this is similar to what Dr. Schomburg found for the males. As their BMI is decreased, their risk increases for age-related macular degeneration. We don't know biologically what this means. There could be something going on with hormones or other risk factors that we can't measure, but this is what we're finding. And we have to replicate this. And we're running these markers right now in our Korean cohort, because that's a large cohort. It's almost 1,500 individuals. But we're very excited by this finding. We didn't have this ability to run these statistical programs a few years ago when we had these tools. Next slide. The thing is, we can't look at these factors in isolation, and we have to run a pathway analysis. And I showed you we have these other modifying genes. And one thing Dr. Hayter and I have done, and she's done it with her mice findings, and we've done it with our human findings, is we do a pathway-based analysis. And we say, we have vitamin D, and we have these other genetic findings. How do they all work together? Well, I told you we have these other modifying genes. We know that ROBO and MURA, and we've published this, also work with the vitamin D genes. Here we have the vitamin D receptor gene, and we have the major catabolizing gene of the vitamin D pathway, and we have vitamin D3 over here. Well, these also have a network that also integrate with the vitamin D pathway. So you also wanted to see if these genes were interacting with our other genetic findings. Next slide, please. So we also do other tests for statistical interactions, not just BMI and vitamin D, because we have to think of modifiers and we have to think of possible therapeutic targets. But lo and behold, we didn't find any other interactions. We also want to think of major players, because as yet, even though we know how complement factor H functions, we still don't have a function for ARMS2 HTRA, and we still don't have a pathway yet for this gene or genes. Next slide. So in conclusion, we have found a significant interaction between body mass index and SNPs in the major catabolizing gene of vitamin D after correction from multiple testing. Replication of this interaction is ongoing, and this adds to the previously published literature for a role in vitamin D metabolism and body mass index separately for their association with pathophysiology of advanced age-related macular degeneration. Next slide, please. And no researcher is an island. None of this work would be possible without the people in my laboratory. Dr. Hartnett, who's involved now also in a lot of the phenotyping, Dr. Ivana Kim, Dr. Joan Miller at Massachusetts Eye and Ear Infirmary, Dr. Nina Haydar, our wonderful collaborators at Seoul National University, Lindsay Farrer, who also helps us with the statistics at Boston University, collaborators at University Thessaly School of Medicine, and Julie Silvestri at Queen's University in Belfast, and funding from the NEI, the Research to Prevent Blindness, and the Al Sam Foundation. Thank you so much.